By now, you should have a good grasp of what evolution is. This video is about G.H. Hardy and Wilhelm Weinberg, two scientists who took evolution to a new level by describing populations that aren't evolving. This is video 2.3 on the Hardy-Weinberg equilibrium model. The video covers these goals. Pause for a closer look. We've spent so much time thinking about how populations evolve, and Hardy and Weinberg flipped that idea and explained how evolution doesn't happen. They outlined 5 conditions for genetic equilibrium. Here's the list. For a population to be in genetic equilibrium, or stable and not evolving, the population should have a large gene pool, the population should have no gene flow, no mutations, and no natural selection. Finally, breeders in the population should have random mating. Let me explain each of those in a little more detail. First, it's important to explain what a gene pool is. A gene pool is the collection of all of the alleles in a population at one time. And you can measure a gene pool by looking at allelic frequencies. Let's unpack that a little bit. In your first biology class, you should have done Punnett squares like this. Now, alleles are those letters in Punnett squares. Every organism has 2 letters, or 2 alleles, one from each parent. And a gene pool is like taking all of the allele letters from all of the animals in the population and putting them in a big pile, or a pool like this picture right here. Now, that big pile is the gene pool. And the allelic frequency is the proportion of the big letter in here, all of the big letters to all of the small letters, or all of the dominant alleles to the recessive alleles. So why do we need a large gene pool for a population to be stable? Well, let's imagine 2 gene pools. Now, both gene pools have 75% dominant alleles and 25% recessive alleles. In this small gene pool, there are 3 big R organisms and 1 little r organism. In the large gene pool, there's 3,000 big Rs and 1,000 little r's. Now, what would happen in each population if 1 little r organism died? Well, in the small population, the little r guy dying would have a huge impact, shifting the gene pool from 75% big R to 100% big R and 0% little r. But in the large population, the death of one of these 1,000 little r organisms would change the frequency an insignificant amount, so that no change would occur, and this population would be more stable. So individuals can enter or leave large populations without a big impact to the allele frequencies. An individual that enters or leaves a small population has a big impact that can change the population and cause evolution. Next, for a population to be in equilibrium, there must be no gene flow. Gene flow is immigration and emigration, where organisms enter or leave a population. Individuals leaving take their alleles out of the gene pool, and individuals entering a population add their alleles to the gene pool. Either way, gene flow can change the allelic frequency, so stability requires no gene flow. Next is no mutations. Mutations can cause evolution by introducing new variations, like this red bird right here. And since mutations introduce change, mutations cause evolution. A stable population in Hardy-Weinberg equilibrium has no mutations. Hopefully, no natural selection is really obvious. Natural selection is a process that causes evolution, so a population in equilibrium can't have natural selection. Hopefully you're seeing how this equilibrium is more of an idea than a real thing. Finally, mating must be random to have equilibrium, where organisms pick mates randomly and not based on any fitness characteristics. Here's an example down here. In random mating, this dark brown beetle would mate with equal chance to another dark brown beetle, a light brown beetle, or a medium brown beetle. In non-random mating, maybe the dark brown beetle could only mate with other dark brown variants or something like that. And then with this selective breeding of dark brown with dark brown, the population would evolve so that dark brown is more represented. So to have a non-evolving population in equilibrium, there must be random mating and no sexual selection. If all 5 of these conditions are met, and they usually are not in the real natural world, 2 math equations are unlocked that can describe the stable population. P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1. These are the Hardy-Weinberg equations. Let's use a Punnett square to help us understand what's going on up here. Now, a Punnett square only shows one breeding pair, this plant and this plant. These equations, however, take into account every breeder in the whole gene pool. So what do these P's and Q's mean? Well, P and Q are allelic frequencies. Alleles are the versions of a gene. Now, there can be a dominant allele, which is the capital letter, and a recessive allele, which is the lowercase letter. P is the frequency of the dominant allele, and Q is the frequency of the recessive allele. Think of this top equation as the allele equation. P squared, 2pq, and Q squared are genotypes. A genotype is the combination of two alleles an individual has. There are three genotypes. Homozygous dominant, which is big R, that corresponds to P squared. Heterozygous, big R, corresponds to PQ, and homozygous recessive, little r, corresponds to Q squared. So this bottom equation, think of that as the genotype equation. The bottom equation is also used to represent phenotypes. A phenotype is the physiological manifestation of a genotype. In these flowers, imagine that red color is dominant to white color. And this means that big R, and big R genotypes have the dominant phenotype of red. Little r gives the recessive phenotype of white. So, P squared plus 2pq is the frequency of the dominant genotype, red, and Q squared is the frequency of the recessive phenotype, white. In class, we'll do a bunch of examples together, but I'll work one on the video right now. Here it is. Most of the American white population can taste the chemical PTC. This is the dominant phenotype. 9% are non-tasters, the recessive phenotype. Determine the frequency of A, the dominant allele, B, homozygous tasters, and C, heterozygous tasters. I like to think about what's given first. The prompt tells us that 9% of the population is non-tasters, the recessive phenotype. In Hardy-Weinberg problems, we express numbers as decimals, so we got to convert that 9% to 0.09. Also, the recessive phenotype is represented by little r, so let's put that in there too. All right, there we go. And in Hardy-Weinberg symbols, little r is Q squared. So there's that, and we have Q squared equals 0.09. All right, let's simplify that to get rid of all of the extra noise. From here, to find Q, we just need to take the square root of Q squared, or take the square root of 0.09. The square root of 0.09 is 0.3, so Q is equal to 0.3. Now, figure out P by using this top equation. Plug in 0.3 for Q and solve to find P, and that means P is equal to 0.7. And now that we have P and Q, let's reorganize. Much more simple without that other stuff in the way. Now, what's this problem asking for? First is the frequency of the dominant allele. In Hardy-Weinberg symbols, the dominant allele is P, so the frequency of the dominant allele is 0.7. Boom, there it is. Next is the frequency of homozygous tasters. Homozygous tasters have the genotype of big A, which in Hardy-Weinberg symbols is P squared. So there's that right there, and now let's do the math. P squared is 0.7 squared, and that's equal to 0.49. So 0.49 is the answer for part B. Finally is the heterozygous tasters, which would be big A, little a. And in Hardy-Weinberg, the frequency of that is 2pq. Let's do the math. So here we go, 2pq is 2 times 0.7 times 0.3, and that works out here to 0.42. We'll do a bunch of problems in class, and I know with a little bit of practice, you'll catch on. Recap time. This video was about populations in Hardy-Weinberg equilibrium that are genetically stable and not evolving. To be in Hardy-Weinberg equilibrium, populations have to meet all 5 of these conditions. Memorize them. And the crazy thing is that this pretty much never happens in nature. The Hardy-Weinberg model includes math equations that we could use to calculate the frequencies of alleles and genotypes and phenotypes of genetically stable populations. The usefulness of this will become more clear later on.